C'est bon? Ouais. Bon, ouais. Bonjour à tous et à toutes, merci d'être venus. Donc aujourd'hui, on va accueillir Ralph, qui est professeur à l'Université fédérale de Pernambuco à Recife, au Brésil. Il va vous présenter un travail sur les size spectra et je vais le laisser se présenter.
Bon, bonjour à tous et à toutes, mais je crois qu'on va faire ça en anglais, non? So, thank you so much for the invitation, especially to Arnaud, Otipa, and everybody, and I'm very glad to be here again after about 1.5 years and to present something about size spectra again after last time we were talking mostly about data and results. We will today talk a little bit more about theory and ideas, not so much about data and results. Although I will also present very shortly some results that we also have. We are going to address some recent efforts towards a complete theory of scale-invariant ecosystem size spectra. So we're going to talk about these efforts, these ideas, these advances in concepts and theory. And we're also going to talk about what we are actually sampling, or what is biomass. Most of you think you know what is biomass. You have been sampling biomass maybe all your life, but actually it's not as simple as we always think it is. So first, a very brief introduction. What are size spectra? Most of you know, but just briefly, it's the relationship between body size or body mass and abundance or total biomass. It's just a way to describe an ecosystem and to show that there are lots of very small organisms and there are very few of the large ones. So you know, obviously, that if you have an ecosystem, you have very few large whales, large fish, and so on. And if you take a little drop of water, you have 10,000s of cells of phytoplankton. So the small ones are very frequent, very abundant. And they follow what we call the power law of Pareto distribution. There's a lot of the small organisms, very few of the large ones. And when we plot this on a log-log transformation, we get this usually nicely linear shape, which often has a slope of -1, which is also intriguing. Why -1? Why is it so constant and ubiquitous everywhere? So the observation that we have a constant slope, the log-log transform, means that we have the same processes and the same laws and the same things going on independently of scale. That means that for the small organisms, the unicellular ones, the mesozooplankton, the fish, and also the much larger organisms, we have some kind of set of laws that govern natural ecosystems across all scales. The same kind of relationships, the same kind of general laws apply to the small, the medium, and the large organisms. And we don't know which laws. So we are still seeking for a universal law of ecosystems on our planet. And it's a bit like the fractals. It sounds a little bit like fractals. If you remember what fractals are these geometric forms, that are very beautiful. And when you zoom in, you have exactly the same shape. The same forms appear wherever you zoom in. Even in the microscopic things and the large things, you have this sort of a little bit analogous to fractals. Remember that. So we have scale-invariant processes and laws that we are searching. There's a search for this. And so we need this complete theory across the complete ecosystem. And you maybe wondered why I wrote this like this, complete with E-A-T. It's not a spelling error, because if you put this into a spell check, like in Grammarly or so, they always complain, they always want to fix that, but it's actually on purpose. It's a nod to the life work of Daniel Pauly. Maybe some of you remember, the first complete elephant was also written like this. And at that time, I was talking to Daniel Pauly and asked him, that's a typo, right? That's not, no, it's on purpose. And complete means they're all elements and they fit nicely together. So it means all elements are there and they work nicely together. That's what complete means. And it's a nod to the life work of Daniel Pauly. And it's also the attempt to combine size spectra theory and length-based fishery science, which have been apart for a long time. No, it's also a way to combine the things that I'm working with, also to, let's say, organize the mess that my scientific life is into one single thing, which is the combination of the plankton work, which is mostly size spectra, and the fishery science work. Like the work on elephant, FISA2, tropefish R, and most recently on fish boot. And now I will just make a short advertisement of the fish boot workshop in September. It's online. If you want to participate, we have this fish boot package, the software package in September, our workshop. So it's we need this concise, unified general theory of ecosystems. But then I told this to Daniel Pauly one day, I think it was 2018, during a workshop, a length-based fisheries workshop in Bremen, 2018, and I sat with him again, like talking in the middle of some coffee or so, and so, well, I think we should try to get finally this unified general theory. And well, he said it's completely unnecessary. It's something that doesn't make sense. It's not possible. It's impossible, and it doesn't make sense, and it's not necessary. And he told me explicitly, you shouldn't do that. You shouldn't lose your time with such a stuff. No, and that's when I started working more intensively on the quest of a unified theory. Although I've been working like about 20 or 30 years, more or less on this. But after this, I started working more intensively since 2018. And maybe it's not that unnecessary. Maybe we can answer a few questions and solve a few paradoxes with a unified theory. For example, a paradox that has been debated for many decades, which is the paradox between the very constant and invariant size spectra, which are almost approximately -1 in the slope, which are always very nicely, whether you go polar regions, tropical, upwelling, everywhere, where you go for the phytoplankton, zooplankton, fish, whatever, you always get this. It's very constant, but trophic pyramids, it's a mess. They're completely messy. You have top down, you have wasp waves, you have hourglass shapes, and the inverted pyramids is not an exception. We've been mapping pyramids like for a few years in our working groups, and about half of the trophic pyramids on our planet are inverted. So trophic pyramids are really a mess, very variable in size spectra. On the same planet, on our planet, we have these completely different situations. So maybe we can answer this paradox using some ideas from these unified theory efforts. So a new conceptual framework is needed to solve the size spectra trophic pyramids paradox and many other paradoxes and questions that we can answer with the unified theory. Is it actually -1? Well, we have a lot of data, just to show very briefly. For example, here from the Abraços and Tapioca project, I have to show this. It's obligatory. I have to show some results from Tapioca and Abraços and show these beautiful diagrams. So we had some results with size spectra, for example, here. The zooplankton size spectra from the tropical Atlantic is approximately -1 in the biovolume biomass spectrum, and also in the Triabras project, we got for the complete tropical and South Atlantic, and we also have this nice size spectrum for phytoplankton, zooplankton, and also for the mesopelagic organisms. However, there are many people who don't believe this. We can put so many data as we want. There are many people who say the size spectrum is flat. No? So is there a flat size? It's like the flat Earth people. You can tell whatever data you want. They will always say the size spectrum is flat. So, and actually, it's not like some very irrelevant, crazy people. It's, for example, in the metabolic theory of ecology, which is a very important theory, very widely cited. If you read the key paper of MTE, of metabolic theory of ecology, Brown 2004, it's a flat spectrum. All have the same biomass. Phytoplankton, zooplankton, fish, all the same biomass. So that's where we see always the evenly distributed biomass. So wherever you could look at the internet, you see this, you will come across the evenly distributed biomass. Or some people acknowledge that in nature, it's somehow not flat, but when you project it on log space, then it's evenly distributed in log space. So we will see how this comes in kinds of method, how it comes to the evenly log space. It's a kind of distortion that occurs when using log scaled vectors that we will see in a few minutes. So all this confusion and paradoxes, they call for a unified general theory, and that's where we come to my favorite pets. It's not like our little dogs at home, I mean the theory, and it's like the predator-prey efficiency theory of size spectra. It's available at arXiv. I have the link here somewhere, so you can, or it's a very long text, sorry, it's a lot of stuff, but maybe we will try to publish a short version in the next few months. And also we have this design with these sardines. Why are these sardines there? Because we address questions regarding the maximum biomass in ecosystems. If you see, like sardines have a lot of growth and body growth, population growth, reproduction, so you have a lot of sardines, and if this would go on and on and on, at some point we would have lots of sardines everywhere in the Atlantic, and we would be able to walk here from Set to Brazil. I would like to be able to walk or drive there, but for some reason, we don't have so many sardines here, so something limits the biomass in natural ecosystems. No, is it bottom-up processes, top-down processes? So that's what it is mostly about. So what is PETS? PETS is one simple equation, very simplified, very condensed down to one simple equation, and a set of hypotheses and mechanisms, a list of hypotheses, mechanisms, and testable predictions. The PETS central equilibrium equation is that the slope, this log linear slope of the size spectrum can be explained as the ratio of the mass-specific trophic efficiency, E, and the log of the predator-prey mass ratio. So these are two things that are very simple and also, most importantly, precise in the sense that there is no additional term also. Because it's easy to have such an equation if you have an additional term plus 0.75, plus 0. It's the exact precise units and values. So this gives a prediction of the -1 slope in our general ecosystems. And it's also easy to memorize. So what does it mean? It means that you have predator-prey relationships where you have a high PPMR. That means there's a big difference with very large predators and small prey, like large whales, small krill. And then you have a low efficiency. You have systems where you have very close together the mass, like tuna and sardines. Then you have a low PPMR and high trophic efficiency. That's what it basically means. And the slope here is always -1, and we also actually have a more or less constant intercept. And this also means that we have a scale-invariant relationship between efficiency and predator-prey mass ratio, not the ratio of the mass of the predators and the prey. That means that large predators have a lower efficiency. You see, here's E, the efficiency, here's the predator-prey mass ratio, so large predators have low efficiency. That makes total sense. If you see, for example, the K1 assimilation efficiency works from David Pauly from the 90s that describe assimilation efficiency with size and many other works related to efficiency and size. You see that larger organisms generally have lower efficiency, and PPMR, the variability in PPMR is generally driven by variation in predator size. There's this Barnes et al. 2010 paper that shows this very nicely. So large predators have lower efficiency, and we see this in this equation. So But does the equation solve all our questions and show how ecosystems work? No, it doesn't. It's a descriptive equation. If you imagine like a river, a landscape, it shows the river, it shows the landscape, but it does not give you the mechanisms that shape the river. That, for example, in the case of a river, the turbulence, sediment transport, and so on, that shape a river constantly. So here we just have a descriptive set of equations, but we need a set of hypotheses and mechanisms that help us understand how this equilibrium is maintained in a dynamic constant. So we need a set of dynamic processes that we have to understand and try to investigate to describe this. So the PETS theory has in its core this simple equilibrium equation and a set of hypotheses and mechanisms that you're seeing here, and also a set of size boundaries on the spectrum. We just said that the spectrum is based on size invariant, scale invariant laws, but still we have a set of size boundaries that I will explain in a few minutes. We have a set of environmental drivers, biological drivers, very important: metabolism, oxygen, food abundance, nutrient flux, and temperature as drivers in the system. And we have a set of human pressures, for example, overfishing and microplastics. And we have also in our working group a lot of research, lab research, experiments going on the effects of microplastics on size spectra and natural systems at UFPE. So this is also an area of research in our working group. So what stabilizing mechanisms can we imagine? Metabolism, changes in metabolism with size, it's well known. Top-down and bottom-up processes are the main stabilizing mechanism, and also something that we have been working on for the past 10, 20 years are size niche processes, where size niches are dynamically occupied, which has to do with neutral theory and stochastic processes. That means that within each size niche, you can have different species, different taxa, different larval stages, different organisms occupying. Stochastically, that means by chance, the different organisms will dominate within each size niche, but the spectrum is deterministic. It's stable. So our stabilizing mechanisms, they stabilize the size spectrum, but they do not stabilize the trophic level biomass relationships. That's why we come back to our size spectra trophic pyramids paradox. That means that the prey and predator relationships, they have an influence on size, but not on trophic level. It's very simple to understand. The predators, they care about the prey size, not about the prey trophic level. The predators, let's say the tuna or sharks or whatever, they don't care whether the prey has trophic level 2, 3, 4, whatever. They go, selection is mainly by size, food particle size, and so on. That's why the mechanisms that we are talking about, they stabilize on size, but not on trophic level. So, is that all new? Did I just invent this a few days ago? No, of course not. Predator-prey equilibrium theories of size spectra are not new. The first attempt to do something very preliminary from nowadays perspective was from Kerr, 1944. Then Bergman in 1987 presented a very important advancement of our understanding of trophic efficiency in terms of the size spectrum slope. Then the Zhu 2006 paper is widely cited in the plankton literature. Then we have the size-specific trophic models, which have contributed a lot to our understanding of size spectra. For example, the apicosm of Olivier Maury and contributors. I don't know whether Olivier Maury is online. And also the MICE software has also contributed a lot to this. And then we have the Anderson and Bayer, Anderson et al., Anderson and Peterson works, all very important. The Barnes et al. 2010, where predator size and PPMR are linked, is very important in this context. And now we are at the PETS, where we have the simplified equation and this short list of testable hypotheses and predictions. So this is a very, it's a short summary of the history, of equilibrium size spectra theory. So now we come to the mechanisms. I will not show all mechanisms, it's a long list, but let's focus on a few simple equilibrium mechanisms that lead to a stable spectrum. For example, the rise TOPS mechanisms. Rise means replacement in size structured ecosystems. It's a mechanism for compensating for gaps. That means when you have a gap or a hole in the spectrum, there's a mechanism that will fill in the gap and maintain it stable. No, it's the gap filling. Like if you have a road that's full of someone goes there and fills the gaps. That's what rise is about. And the TOPS mechanisms or TOPS hypothesis is a top-down overshoot peak suppression where it controls positive deviations. So when there's some outflow, some peaks, it goes on the peaks and gets the peaks away from the spectrum. So let's have a look at the TOPS mechanism. No, top-down overshoot peak suppression. It's biomass overshoot suppression and maintains a constant biomass. It's very easy to understand, very easy to imagine. Imagine yourself a savanna somewhere in Africa, and you have a few zebras like looking out over the high grass. And then you have more and more zebras and a lot of zebra biomass. The predators will take notice of this. If you have a lot of biomass there, predators will take notice and they will change their behavior instantly. So that's basically what it is about. It's the suppression of the biomass through top-down processes. And in the size spectrum context, we have this peak, then we have the increased predation, overshoot suppression, and the restored spectrum. Very simple, very simplistic, very easy to understand. And we can also describe this like as a lawn mower, where you have the consumers going to the peaks, you have lower biomass coming to the carrying capacity spectrum, and then we'll go here. So it's very difficult to observe because the dynamic process, we have these what called terradynamics, these stationary trophic cascade waves, as we call them. Nature is more complex, they are not lawnmowers, and we have the defense mechanism, so in fact it's much more complex and very difficult to observe in nature, although it's conceptually simplistic. It's very difficult to observe in nature. And now we go here for a real example, like a bloom in a phytoplankton bloom. You have the disturbed spectrum with the bloom, and then you have the restored spectrum. But what makes a bloom collapse? There's a lot of work on this from Xavier Irigoyen and many theories and so, and many observations. But basically, it's increased raising, overshoot suppression, but also other factors like the sinking of the bloom, the sinking of the phytoplankton. Light is important. Nutrient deficits appear at the end of the blooms. Oxygen at night, you have oxygen deficiency, and also you have the problems related to ammonia, specifically at night. So it's more complex than that. Another situation is when you have in upwelling systems a lot of nutrients coming in all the time. When you have the nutrient flux all the time, the nutrients, you have high phytoplankton biomass, and of course this biomass is transferred to the upper trophic levels via the zooplankton, so the zooplankton biomass increases. And so in an upwelling spectrum, we would expect the phytoplankton biomass to increase, but specifically the zooplankton biomass to increase a lot by many orders of magnitude, but still keeping the -1 spectrum. So that's what we observed in the Benguela upwelling system, the most productive upwelling system on our planet, where we had exactly this high phytoplankton biomass of the nano and microphytoplankton, and very high, many orders of magnitude, crustacean zooplankton biomass. So this is really an interesting observation from the TriAtlas project. The other hypothesis, the RAP hypothesis, replacement in size structured ecosystems, is about filling the gaps and the growth. You have, for example, a mass mortality event. Then you have a gap or a growth in the spectrum, and it's restored because of density dependent mortality, increased growth, and so on. And then you get a bottom up cascade wave. So the biomass goes somewhere here to the cascade wave. Another scenario is where you have local extinction, then another taxonomic group will replace, and you will get back to the original. So it's about gap filling. You know, it's a lot of theory, functional replacement, size niche, occupation. That's what RISE is about. Sometimes we have continuous invasions, for example, exotic species. Don't think about lionfish and many other exotic species you have. And then you have a continuous replacement and the original spectrum is restored. This is theory, but we also have observations. I will not, we can talk hours about our time series observations in our paper. So I refer to our paper here from last year, where we have the size niche approach implemented from observations. For example, if we have in the estuary, we also have working on the shelf in the coastal bay area of Brazil, near Recife. But let's look at the estuary. In the estuary, you have the mangroves with lots of invertebrates, for example, crabs. A huge biomass of crabs that most of you know about, the crabs. And the crabs, they produce larvae, microscopic zooplankton larvae. And at some point, these larvae are released into the estuary. So that's a nice experiment, no? I mean, I can't do this in the lab, but the nature makes this nice experiment for me, producing lots of larvae and launching them into the estuary. And what happens when the crab larvae are released in huge biomass into the system? They replace the copepods. So that's what we see here. We see that this is the size spectrum here in the estuary, one example of one situation in the estuary. The green lines here are the copepods. The orange lines here are the crab larvae. And you see that the crab larvae have a peak just in the middle of the copepods. And the overall spectrum is this black line. So you see they're empty bins where the crab larvae have made a hole or a gap into the copepod spectrum. We usually have this nice copepod spectrum, and the crabs have gone into there and replaced and displaced the copepods here. So whether it's predation or competition, so we can speculate, but no doubt there have been a replacement of copepods by the crabs in this situation. So this is an example of functional replacement. Just very briefly, we don't have so much time. So our key mechanisms that we just have learned, two of them, is the TOPS, which is the lawnmower, which you see there, and RISE, which stabilizes the continuity of the spectrum. No, it's gap filling, because you have this continuous nice spectrum, there are no holes in it. This is about the RISE mechanism. But we have a third one, we have a lot, but I will just focus on these three key mechanisms, which is TOPS, RISE, and SOFT, which is the size spectrum specific optimal foraging theory. So it's based on optimal foraging theory that many of you know about. It's been published 20 or 30 or 40 years ago already. But this is the optimal foraging theory within the context of the size spectrum. So how do the predators affect the size spectrum, and how does changes in the size spectrum, the size distributions affect the behavior and the size selectivity of the predators? So this is the soft theory, which stabilizes the size spectrum slope. So here we have removing overshoots. When you have, like, the grass is too high, it overshoots. The rise is where you have the gaps, you're filling the gaps, and the soft theory stabilizes the slope on the -1. And how does it work? It's also a long part, but I'm going to summarize it. It's based on trophic cascade waves. So you have bottom-up cascade waves and top-down cascade waves, and the way that these cascade waves act on the spectrum stabilizes the spectrum based on the response of the predators to changes in the size spectrum, of the relative composition of small and large food and their size selectivity within these cascade waves. So I refer to the paper, but it would be too long here just to describe both the top-down and the bottom-up cascade waves in the context of the soft mechanisms. And the next point is the maximum carrying capacity, which was also something that appeared in the data, and we just looked at that, and it's really very interesting to see that you have these low biomass situations in nutrient-stressed spectral situations where you have very low biomass, and then it reaches a ceiling, a maximum, a ceiling. So this ceiling is the maximum carrying capacity. And we saw this also very nicely in the phytoplankton data from the tropical Atlantic and also the mesopelagic fishes in all regions that we have this maximum biomass apparently. And the maximum carrying capacity, while these nutrient-limited spectra, they are all massive, the maximum carrying capacity spectrum, that's the nice one. That's the slope -1. That's where it comes from. The -1 slope nice spectrum is the maximum carrying capacity spectrum. So I talked about the size boundaries. Which size boundaries can we expect in the spectrum? It's where something really important changes in terms of metabolism, predator-prey relationship. So one very clear boundary is the Kleiber SB, Kleiber's M metabolic scaling size boundary. The Kleiber metabolic scaling is the metabolic scaling of eukaryotes like us, cells, no, and that end where we change from the eukaryotes to the prokaryotes, like bacteria, people, prokaryotes, and they have a completely different metabolism size relationship. So that's probably why we have a much steeper spectrum. So we have a lot of bacteria and a lot of picoplankton under certain situations. The other one for the larger organism is the crit-gill boundary. It's more or less the nekton versus plankton boundary. That's where we start having organisms with gills, like fish and mammals, larger organisms. And there is a critical size where organisms start having gills, which has to do, of course, with the surface area relationship. And this can be explained by the GOLT theory of Daniel Paul, the gill oxygen limitation theory. Why do small organisms don't need gills, like copepods don't have gills, but the larger organisms like fish and others that are about more or less larger than approximately 1 centimeter, they all have gills because the surface area ratio gets bad and they need to have more surface. So this is where this changes, and that's also where the growth changes. Here you have exponential growth. Here you have the famous von Bertalanffy growth, which is a completely different form of growth equations, this boundary. And then we have the top dosh, which is the boundary where we have suddenly the crustacean grazing stops being important, which is the top-down controlled size horizon. Why did I call it horizon? Because it sounds nicely and it remembers black holes. I don't know whether you like science fiction and physics and astrophysics, but like black holes, they have a horizon, an information horizon where information cannot leave the black hole. That's the horizon where the top-down cascade waves cannot go and transmit more information to the smaller organisms. That's a horizon where the cascade wave information stops. In theory, we don't know that this is just a theoretical concept, but basically it's based on the idea that crustacean grazers cannot effectively control very, very small prey beyond this boundary, which is more or less similar to the picoplankton, nanoplankton boundary. When you go to the picoplankton, you have mostly protozoan grazing affecting and controlling the spectrum. When you go to the nano and microplankton, you have mostly metazoan grazing affecting the size spectrum, like crustacean grazers, like copepods and euphausids. So we can say it's the boundary where we go in a transition, looking at the phytoplankton spectrum, large phytoplankton, where we have an effect of copepods and euphausids, and the very small phytoplankton where protozoan grazers like ciliates and flagellates are more important, and which have a different growth and metabolism ratios. So something should change there. Here's the critical boundary that I talked about, which is about the slow growth slow asymptotic growth, the von Bertalanffy growth for the larger organisms, and exponential growth for the small ones. And it's related to the Daniel Pauli's GOLT, Gill oxygen limitation theory. So if you would invite Daniel Pauly these days or in the past 10 years, he would probably not talk about the long list of achievements. He always likes to talk about the GOLT theory. And so what is the complete spectrum that I have just talked about? It's where we have all the organisms from the phytoplankton, zooplankton, and fish, and the particles. Why is it important to have the particles? Because particles are also food, and they are also part of the food web. So, and also they appear in the UVP data, so we have to include them. And that's not a new idea. It's not something that I invented. There was also this Seaman and Boss paper and other papers that have suggested that we do need to include particles. And of course, the idea to include detritus particles in trophic modeling is not new at all. It's like since the 60s and 70s. But size spectral science usually does not include particles, only organisms. So we have so many different particles, so many different processes like fisheries. We have to include fishery science, length-based methods in fisheries, and to get into the complete spectrum. So this is all very nice in theory, but what about the methods that we need to generate these spectrum? Where do we get to have these data? We need to have a reliable concept, for example, of biomass and abundance. So here we come to the second part of the talk, which is about what is biomass. Most of you when I would ask you, you would immediately be able to answer this very quickly. Everyone has an idea what biomass is, has been sampling, for example, benthos with a sieve or plankton with a plankton net. Everyone knows what biomass is. But it's not that simple, because our estimate of biomass is a function of the target size and of the size range. That means we have a Pareto distribution, many small organisms, very few large ones. If I choose smaller organisms, I have more biomass. If I choose larger organisms, I have very few biomass. Everybody who knows that, we have been working with plankton nets so many years, like 30 or 40 years in our group, with so many different plankton nets, and the small mesh plankton nets, like 50 micron, 60 micron, there's a lot of biomass. It's like a thick soup with so much biomass. And the larger nets, like 500 micron biomass, is just a very little amount of biomass. It's really obvious. No? And we also have a size range. If we get a wider size range, we have more biomass. A smaller size range will give us a smaller biomass estimate. And also, if I go here for the limits, upper and lower limits, you start understanding that we are always within a certain sector or range or bin. Well, these are bins. I have to go for a bin. But what is biomass? Is it the area under the curve, or is it the mean y value here in the curve? So what is actually biomass here? Maybe it's impossible to determine actual biomass because it's based on subjective choices. If I choose a certain net, I will get a biomass. If I choose a certain range, it depends on my choice. So maybe it's impossible to determine biomass because it's so subjective, and we don't understand what biomass actually is. And another problem related to this, in the size spectral science, we usually use the NBSS, which is a normalized biomass size spectrum. And it has such weird units. You see here, grams, carbon per square meter, and so on. It doesn't look like a biomass. I don't think it's biomass. Actually, it's not biomass. So the normalized biomass size spectrum is probably not biomass. And you see at the units here, grams carbon per cubic meter divided by grams carbon per individual. This is not a biomass. It's biomass divided by body mass. Why is that so? Because normalizing means dividing by bin width. It's the biomass divided by bin width. So it's not something that I just discovered yesterday. There are lots of papers about this, also since the 80s. And in the Tri-Atlas working group, we had so many discussions about this. We have colleagues who think it's abundance, like the people from Ecosum friends or people from Defonge and others. We had so many discussions what this is actually about, what is the units, what is the dimensions, what does it represent. And if you look at the units, you can simplify this, no? Grams per cubic meter divided by grams per individual. Then you can simplify this. The grams cancel each other out, no, here. And then you get individuals per cubic meter. So actually the NBSS has abundance units. It's a biomass spectrum, no, normalized biomass divided by bin width and has abundance units. So there's this discussion. Is it actually, is our size spectrum? Because that's what we work with. The NBSS is what we have on our table. That's our data. That's what we have to work with. Is the NBSS actually abundance? Is it biomass flux? Many authors in the 70s and 80s have said it's biomass flux. So the NBSS represents the flux of biomass through the spectrum, how fast the biomass goes from the small to the bigger ones. Is it biomass flux? Or is it really biomass? What is the NBSS? What is biomass? So let's go back to the basics. It's very confusing, maybe, no? So let's go back to the basics. Very basic, no? We have the large body size with very rare organisms. Very few, very rare. Like tuna, so they're very rare. The small body size, they are very frequent. This is a simulation of a Pareto distribution. No? Here. You see the small ones, you have a lot of them, high biomass. So the large are very rare, the small ones are very frequent. You have a lot of them. High abundance, high biomass in the small organisms. And then we will try, what would you try to do usually to describe a size distribution? Is to do a histogram. What is a histogram? You have the size classes and then put your size class. So since we all have these blue small ones, the biomass is all in the first bin. The first bin or the first column is where all the biomass. So this is a really bad histogram. I cannot see a shape or distribution, I just see one column. So I go for a log axis still. Most of it here is seen in the first one, and we have gaps. There's a gap here. 0 observations, also bad. So that's why we go for irregular log-scaled histograms. We don't use in size spectral science regularly spaced histograms. We use the ones that are log scale, where you have a very small bin width here, and the bin width increases logarithmically or geometrically to get rid of this. But this gives us a distortion of the size spectrum. This is what we are going to see in a few minutes. So this is a big problem, no? So another problem is then that the Pareto distribution or power law distribution has no defined mean, mode, or mean. There's no mean or mode or whatever. So you know the common normal distribution. The normal distribution that we learned in our first semester in statistics, normal distribution is really nice. Everybody knows, no? The mode, so beautiful, no? And then we have the mean and the median. It's so wonderful. But it's completely useless in ecology because we never have the normal distribution, as you know. And in the Pareto power law distribution, if you have such a distribution, you don't have a defined mode and also no defined mean or median. Instead, we have to describe it in a different way with these sectors or histograms. But then we come with these sectors and we see this area. Is the area the biomass? Is the y value the biomass? So I will say the area maybe is the biomass. But if the area is the biomass, what is the y value? Is this biomass? And it cannot be biomass both on the y and here. So what is that all about? No? So first, there's no objective estimate of biomass. There's no consistent theory regarding what these integrals or whatever are about. Because when I see area, I think integral. Is it an integral? Or is it just a bent approximation of the shape? And it's maybe not an integral. So what is it all about? Let us have a look at this conceptual graph of the cumulative biomass distribution, where it all begins. So the cumulative biomass distribution gives us an idea of how fast biomass accumulates when we increase with size. The cumulative biomass distribution has a defined first derivative, which is the biomass density distribution. So here we have a nice, mathematically beautiful, clean derivative integral relationship, like you have learned in high school or the first semester somewhere. No, this is a really nice math with a derivative and integral between the cumulative biomass distribution and the biomass density distribution. But this is not real biomass. This is the biomass density distribution. D describes how biomass declines with mass. So D has the units of per cubic meters, not bio. The biomass is a binned approximation of D. So we have to do the binning. When we have the binning, we have these nice squares, and the binned approximation of D is the binned biomass B. So this is biomass. Here we have the biomass. And the biomass, because it's an area, it has these nice biomass units. This is really, now everybody, I see some people smiling, being happy. So finally we got into nice units and nice biomass. And in practice, we see that D and D have the same slope, the same shape, no? But they have different intercepts because since it's a binned approximation, if I have larger bins, then I have more biomass. So the intercept here is a function of the bin width. Larger bins, more biomass, but the shape is the same. So the biomass is an approximation of the density distribution, and the density distribution is also an approximation. We can represent the shape and the slope, but not the intercept. So the NBSS, coming back to our practical things, we do NBSS when we do size spectra theory. When we want to obtain a size spectrum in nature, we do this here, NBSS. But what does it represent? The NBSS is a representation of D, of the biomass density distribution in units per cubic meter. That's what we do when we do an NBSS. But I would like to have this one. I really like biomass. I have always worked with biomass, so I want to have a representation of biomass, and the representation of biomass in the spectrum is urgently needed, and we want to have this. So I propose this new BNBS method, the back-transformed and normalized biomass spectrum that has nice, convenient biomass units, no gram per cubic meter. So here we have a size spectrum science with nice biomass units and dimensions, and we can see here where we are with the VNBS, the common NBSS that we have been working, and how do we get there in terms of transformations and math. We have the biomass density spectrum, D, that we saw in nature, that is impossible to observe. It's impossible to obtain the biomass density spectrum in nature. Once we get a sample, we are here, we have these bins. But usually we do the binning of the biomass density spectrum with log scale bins. And the log scale bins, because they're small and large and so on, they have a consequence, which is the transformation and deformation of the spectrum into a flat BWID spectrum. Well, BWB is the bin width inflated biomass. The bin width inflated biomass spectrum is the D flat, and some people think that's the spectrum. Really, there are many people who think that's the correct way to represent the biomass spectrum. For example, last year in September, when I was on the Azores in a workshop at Atlantic, I had a very long, almost 1 or 2 hours argument after my talk, where there was an Italian physicist who was on microplastics, who was very emphatically defending this. He said, "Oh, this is wrong. The right thing is this. That's the spectrum, the flat spectrum. The spectrum is flat. You have the same biomass of larger and smaller." I mean, it's obvious who has ever pulled 2 plankton nets that the smaller plankton net has much more biomass. I mean, it's no doubt at all in this. And this is actually a artifact of the binning. The smaller bins, they get less biomass. The larger bins, they get more biomass, and this gives you this distorted spectrum. So they are working on a distorted spectrum. And but this, in this context, is an intermediary step. The bin width inflated biomass is then normalized. We get the NBSS. That's what we usually do. That's what we are doing since 10 or 20 years, the NBSS. And now I'm suggesting to do the back transform into the back transformed normalized biomass spectrum, which has the same slope and the same shape as the NBSS, but has nice convenient biomass units, and where the intercept is different from here, but the slope is the same. So this is about a summary what I have been proposing recently. And also this BMBS method has these nice statistically robust bootstrap in a two-step bootstrap, because usually people use these points, 3 points, and make the statistics as if it was just 3 points. But actually each point is based on a lot of organisms. So you have to get the uncertainty from the original sampling into the degrees of freedom and make a proper two-step bootstrap. So this is also a statistically robust approach to BMBS. So this BMBS and NBSS story is also available here at arXiv, and it's going to be submitted for publication soon. And just finishing the talk, from the PETs, we have this framework for a systematic discussion, because it's a process. We're working on this. No, and this is intended. Why I'm doing this talk here? Because it's a stimulus for more research on size spectra. So I want you to think and invest time into research in size spectra and participate in our quest for unified theory. So thank you very much for your attention.
Thank you very much, Harald. Are there any questions, des questions? Maybe online too? I don't know. Okay. Also, I have some questions. For the first part of your talk, you talked about several hypotheses of how disturbances in size were adjusted by nature and natural mechanisms. But is there some thresholds where some mechanisms some disturbances, can't be adjusted?
So the question is about a threshold for catastrophic breakdown of the system.
Mm-hmm.
Yeah, that's a very important question, and has to do with the effect of higher trophic levels and larger organisms on the size spectrum, which has to do with the trophic cascade waves, how they propagate, and how they affect the whole size spectrum. So there are a lot of possible, let's say, models and hypotheses regarding this. One is that we may get a greener planet with more phytoplankton because of the larger organisms removed, and the intermediate ones get a higher biomass, and then the herbivores get a lower biomass, and then we have a greener phytoplankton. But so as an example how complex this is in the cascade waves, but it's not easily answered, this question. It has to do, of course, with the cascade waves.
Thank you.
And it's a wide area of research, and it has to do, of course, with such things as a balanced harvesting. There has been this proposal for harvesting small or medium and large-sized organisms instead of focusing only on the large ones. And it's a lot of things that we can discuss about this. I personally don't like so much balanced harvesting, because some people use it as a pretext to fish more intensively, use smaller mesh size, or even do whaling. There are people who do whaling and say that they do balanced harvesting. So balanced harvesting has been used as a pretext for all kinds of things. But thank you so much for the question.
I also have a question on this part. Tu peux te rapprocher prendre le micro pour que les gens en ligne
Te mettre ici, comme ça.
I also question on this part. You didn't speak about time. Sometimes it can be long to reach equilibrium, the new equilibrium. So is there a way to incorporate time in this theory?
Yeah, that's a very good point. Actually, time has been an essential part of size spectral theory from the beginning. The very, very first size spectral theories from Platt and Denman from 77 and 78, they focus on the time. And that's very important. Why? Because, of course, the smaller organisms have a much faster turnover time than larger ones. And this also explains, maybe in part, why we have the damped cascade waves. What are damped cascade waves? That means that you have a lot of oscillations when you have a disturbance, like fisheries, for example, in the larger organisms. And when you get to the smaller ones, it gets damped. That means you have a long oscillation. When you go to the zooplankton, phytoplankton, it gets somehow lost. And that's what the turnover time is. So it's very, very important. For example, when we go for large organisms like blue whales in the Antarctic, for example, there we have a disturbance that has occurred for many centuries. Very bad disturbance, and many populations in the Antarctic of blue whales have not yet recovered, even since whaling has stopped for many decades. On the other point, you have for phytoplankton, for example, you have reactions to changes in nutrients, which can happen in a few hours. So it's really, very important, this question of time.
It's more a comment than a question, but I think that the time is important. And I think that sometimes it's good to take the time because we are using formula, we are using statistical approaches, but we don't think about, and I like very much what you did because you took the time to think about. And I think that it's an example that we should always have and take the time to think, and we should not use any method because it's fancy or it's commonly used, but what I am doing. And I think that all the questions about biomass, etc., it's basic, but who does? So thank you for that because it's good to take the time.
Thank you so much for your comment. Very nice.
At the beginning of your talk, you were mentioning that you discussed with Daniel Pauly, and he said it was like a unnecessary question, or do you know what was his point? Why does he think it's unnecessary?
That's a good question. I should try to ask him in the next few months or years. But I have the impression that there is a general wall between plankton science and fishery science. And fisheries scientists tend to despise all what has to do with plankton. And size spectral science has been traditionally something of plankton. So maybe that has to do, because fishery science is already so complex and mathematically complex and has so many models, so people don't want to care about these small organisms that are somewhere at the base of the food web. Maybe one can understand this, let's say, from a historical point of view.
Thanks. Nico, tu peux te rapprocher aussi d'un micro, s'il te plaît?
Very small question or remark, but ça marche? You mentioned abundance and biomass, and it seems that it's rather densities of biomass and densities of abundance.
Yes.
Because you are cube meters somewhere, everywhere.
Yeah.
And you never mentioned the term density of
abundance.
Of abundance or of biomass, or is it on purpose or is it?
No, we have, it's a bit complex because density can mean many different things. There are people who avoid using the term density. For example, I have been using densities all the time as density of biomass, as if it was grams per cubic meter, as if this was the density of biomass. But then some people who work also on particles say, no, you cannot use the term density not because of what I said right now, but because density is the density, the specific density that determines the sinking velocity, how much mass is in a particle and determines its sinking. So we got away from using density of biomass in this context. We just say biomass. We just say biomass and mean the amount of biomass per cubic meter, which is defined, as I just said, from the size limits in the spectrum. This
The idea that you play with integrals and derivatives.
Not really, because the biomass in the histogram is a histogram. I always thought it was the histogram approximation of the biomass density. It's not an integral. I always thought it was. The integral is when you have a. I had to study this a lot again because it was from a long time ago. But an integral usually there are many different forms, but the most common one is when you have a lower limit and you increase the upper limit. That's what an integral is more or less about. But the histogram approximation is when you get little pieces of the distribution. So this is not an integral. And the histogram approximation has more or less the same shape as the original distribution. And that's what our biomass size spectra are.
I have one last question, maybe. Have you, I don't know if you discussed that in your paper, do you have like some broad idea of consequences for management of marine resources in general from your theory?
That's a very good point. That's basic science at the moment. At the moment, well, we don't have a management tool that is derived from this, but we hope in the future that we will improve our management tools to include ecosystem consequences. It has to do, of course, with all the ecosystem-based management and so on. So we try to improve our understanding of the system and the interactions between the elements of the system to get better management tools in the future. But that's still for the future.
There is a comment from Olivier online. He can talk today, but he will try to reach out to you, I think. Olivier Mon.
Happy. And I'm looking forward to this.
Okay.
And that's the natural fisheries management that comes out from that is the one you don't want.
Yeah, the balance harvest.
Balance harvest.
Yes, that could happen. That could happen. That and at the end we will probably, if you go really for the math and the population dynamics and the trophic interactions, we will have some kind of balance harvesting. You're right. Yes.
Okay, other question? No. Thank you very much.
Ouais.
Et merci d'être venu. Et il y a aussi un jeudi Mardec jeudi. Voilà. Tu filmes pour un. Ouais, carrément.